Right, Manuel, thank you very much. And thank you very much, Pascal, for the nice talk. So I'm Alex Kwarteng. I'm also from the Department of Biochemistry and Biotechnology, KNUST. And of course, today I'm talking about the microbiome and the lymphatic filariasis. Lymphatic filariasis, as we all know, is a parasitic disease which is transferred by nematode parasites, particularly Richeria bancrofti and the Brugia species. And what actually happens is that once an individual is bitten by the mosquito carrying the parasite, right, it moves into they migrate into the lymphatics. And then with time, these worms develop into adult worms and produce many young ones or younger ones that we call the microfilaria. And then these microfilaria move into the periphery, which are subsequently picked up by mosquitoes during their feeding process, and the transmission continues. Now, it is interesting to know that in some form of the disease, you could develop something like hydrocele, which is accumulation of fluid in the tunica vaginalis. And this has a huge implication on the fertility or reproductive abilities of the man in this case. Of course, in the case of the man and of course for the woman, you may also develop a lymphedema or elsewhere called as elephantiasis, which is as a result of increased inflammation and high production of vascular endothelial growth factors, and eventually resulting in this lymph stasis. So the lymph is not able to move as it will normally would do. And eventually the patient becomes inactive, unable to move, and affect the quality of their lives. What actually happens is that this disease eventually know that this normally occurs in the tropical regions of the world, and therefore where obviously the environment is very suitable for the transmission dynamics to go on. So you cannot find it maybe in the northern part of America and the other colder regions. In the past, of course, literature shows that there were some in other places like Italy, in Germany, 50, 100 years ago, but today it is not there. All this because of increased environmental changes and strict temperature, as we begin to see as an effect of global warming and other stuff. Now, treatment of the disease is particularly through what we call mass drug administration, where individuals in the region are given tablets called ivermectin, albendazole, or DEC, depending on where they live. And then these drugs particularly target the microfilaria, and then eventually, which means that it has less effect on the adult worm. And that means that periodically, once or twice in a year in endemic regions, individuals living there need to take these drugs. Now, one of the interesting things that happens is that once these drugs become very important in reducing the transmission potential of the pathogen involved, little is being done for individuals who develop the disease, the condition. The strategies that is put in place normally will consider something like hygiene, like washing of the feet and so forth and so on. But this actually does not also provide much help, because you cannot continue washing your feet almost every second, because at the later stage of the disease, it gets very complicated and something needs to be done. Where elsewhere people recommend something like surgery, amputation of the leg, but this may not be very affordable for individuals that live in the region. And therefore, there is a need to understand what actually complicates to this particular disease at the later stage of the disease. Now, we have shown that if you come to Ghana recently, that the disease particularly is found in two main parts of the country, in the northern part and in the southern part of the country. And we believe that this spatial variation could be influenced by other factors that are very important. And that's what our recent paper showed. But one of the interesting things that we know is that in as much as the disease is caused by parasites, we believe that the role of bacterial infections significantly leads to advancing the course of the disease. And this has not fully been investigated, particularly in our part of the region, because we've done quite a number of studies where we've shown the effect of acute attacks. So with acute attacks, I'm talking about a situation where the individual experiences acute pains and his leg becomes swollen, and then the patient becomes inability to move or do anything for maybe 14 days or 2 weeks. And therefore, we thought the possibility to look at the microdiversity that complicate the disease was extremely important. And so my research team in the southern part of Ghana began to do a profiling of the bacterial communities of the LF individuals and to look at the promising insight into the bacterial diversity among the individuals to see whether we could come out with any group of bacterial community which significantly complicates the condition. So for our experimental approach, we have a collection of samples. Of course, we process the sample in the lab, do bacterial isolation, do MALDI-TOF identification, and then look at antimicrobial sensitivity. Now, some of the key things that we found is that we see that among the bacteria communities, in terms of the phylum, you have the phylum, the Firmicutes, as the most prevalent of them, almost 70%. The Acinetobacter, you having almost like a little over 16%, and then so forth and so on. So we saw that the Firmicutes were of a greater proportion in this particular case. We also observed higher bacterial diversity in the wounds. Now, it is important to understand that among these individuals who present with the leg conditions, some of them come with wounds and others do not come with wounds. Now, we saw that there was a massive polymicrobial colonization, which contribute to chronicity of the wounds, as has been indicated. And then we also observed that the phylum representation is inconsistent with normal skin microbiota when you compare to healthy individuals. That means that once the individual gets a lymphedema, the microbial community significantly changes compared to those who do not have the condition. We also observed that facultative anaerobes predominate the bacteria that were obtained. And here we saw that those in wounds, you have high proportions in terms of free relative frequencies compared to, of course, the obligate anaerobes. And these are very important observations. Again, we saw that consistent change in the bacteria numbers was very consistent when you compare those with wounds and those without wounds. This is so because in lymphedema, it's characterized by leg stages. So we have stage 1 to stage 7. And then when you profile, right, looking at those with wounds and those without wounds, the bacterial communities were very similar to a certain extent, and which also tells us something very important. But when you get to something like in stage 4, you see that there's some kind of a decrease in the diversity as obtained, even as you go on a bit further. But there are more things that we are going to do. We are looking forward to doing more assays to identify what are the changes and where the unique consistencies lies in this particular case. Again, we're also looking at the increased resistance of the gram-positive bacteria in the wounds to standard antibiotics. So we saw that in addition to the huge diversity that we observed in our study, there is some increased resistance to gram-positive bacteria, normally with the wounds. This is not very uncommon because most of the individuals in such communities shop around for any medication that can help them to relieve the pain that is associated with the swelling. And therefore, shopping and buying of antibiotics is a very common. And therefore, these kind of resistance that we are observing is something that is normally looked for. But of course, the implication is in terms of the management of the disease. Once you have people buying antibiotics without any much control, what actually happens? So we saw massive resistance to tetracycline, penicillin, chloramphenicol, and so forth and so on. This calls for immediate attention for stakeholders and the government to put up good policy in place to be able to address some of these challenges. Again, we saw higher resistance of gram-positive, of course, also in individuals with non-wounds to standard antibiotics. And therefore, even those without wounds, we continue to see that there's some kind of increased resistance to these bacteria and to antibiotics. And therefore, this is also of a major concern in this respect. Recently, we had come out with a publication where we look at the prevalence of methicillin-resistant Staphylococcus species. And we saw that most of the species, most of the Staphylococcus that were isolated from the patient were highly resistant. And this causes what we call the chronic non-healing wounds. And therefore, most of the individuals with wounds who get lymphedema with wounds takes a long time to heal, even if they will heal. These are some of the things that we've started picking up. In our previous studies, we have also seen that what we call the influence of seasonal variation among the individuals. And so what happens is that during the rainfall season, people get a lot of attacks. And the explanation could obviously be that once they get exposed to environmental microbiota diversity, because most of them walk barefooted and are engaged in farming activities, and therefore when it rains, they tend to have more attacks compared to when during the dry season. So these are some of the few observations we've started picking at the moment, and we continue to do more further sequencing of the isolate that we have, and to be able to do proper biobanking so that we'll be able to investigate this further. And therefore, we look forward to joining hands with other partners around the world who are interested in microbiota. Of course, this platform is very important for us so that we can collaborate to harness our efforts together to be able to understand the diversity in full, to make proper management, to propose proper management strategies for individuals who go through this. So for today, we have been able to show you that secondary bacterial infections is very important in complicating the lymphedema development. We are still going on with our analysis, and there are a lot of things to be done. We observe that high diversity of bacterial populations and increased frequencies of facultative anaerobes were observed among the patients. And then, of course, the increased resistance of commensal bacteria to common antibiotics among these patients, because there's no straightforward management protocol that these people normally adhere to. So they are resort to buying of antibiotics and using it without any control. And I would like to take this opportunity to thank my team, wonderful people. When you go to the communities, they all do very well to help. I love all of them, and they do such a great job. And we also take the opportunity to thank our funders. So we are funded by EDCTP, by the Canadian Institute for Advanced Research, and then, of course, an African Research Network for Neglected Tropical Diseases. And then, of course, KNUST is our university, the best university in West Africa. And of course, we also work very close with the Kumasi Center for Collaborative Research. And then this is where all the action goes. Thank you very much, Manuel, and then my other listeners. And I'm ready for questions and comments.
Thanks a lot, Alex, for this exciting talk. So please don't hesitate to put your questions in the question and answer section. I think we have time for one short question. There's one from the panelists that actually can't put it in the Q&A. So Luis, do you want to ask it live?
Yes, Alex, thank you very much for the talk. So the filaria nematodes, they have bacteria living inside them that is obligatory, which is Wolbachia. And sometimes antibiotics are used to treat filariasis because they kill the bacteria. And there's also the possibility that the immune system reacts against the bacteria. So are you putting these two possibilities as causes for the observations that you're having?
Yeah, thank you very much, Louise. One of the interesting things to observe is that normally in the advanced stage of the disease, most of them might even be negative for the Wolbachia. The Wolbachia you're talking about is the endobacteria that lives within the worm, and most of those at the advanced stages of the condition might even be negative for the worm. The worm might be dead. We believe that is as a result of the death of the worm that releases massive antigens, and this is then reacted to by the host immune response, resulting in a massive inflammation which is not controlled. And so in our condition, in this particular context, most of the patients are negative for the worm and they're negative for the Wolbachia. And therefore, we believe that whatever expansion we are seeing is an onset of immune regulation that have been set off being complicated by external or secondary bacterial infection, which is progressing or increasing the progression of the condition, right?